Hello students, welcome to EPG Pathshala. I am Rajat Oberoi from Vision Medicare Industries, New Delhi. In this lecture, we will discuss about lipids as excipients for drug delivery applications. Paper Product Development Part 1. In this topic, there would be 4 learning objectives. 1. Role of lipids as excipients in drug delivery. 2. Various types of lipids used as excipient in drug delivery. 3. Critical properties of lipids that affect their selection as excipients for drug delivery. Lastly, we will discuss about various lipid-based formulations. So, we will start with introducing the topic. It is widely accepted that 40% of new drug candidates and nearly 90% of molecules in drug discovery pipeline are poorly water-soluble and lipophilic. Often, such drug candidates show great potential for improved therapeutic outcomes. However, they present challenges to the formulation scientist since increased lipophilicity and a corresponding decrease in solubility in physiological media is a major obstacle that reduces the overall bioavailability of many of these drug candidates. In the recent years, formulations composed of lipids as excipients called the lipid-based drug delivery systems have gained much importance because lipid offer the potential for enhancing the bioavailability of lipophilic drug candidates. Lipids have been proved to be an attractive excipient for the formulation of pharmaceuticals, vaccines, nutraceuticals, and diagnostics because A. The biocompatibility and safety of lipids has been well established as they are ubiquitously distributed throughout body and play a vital role in the functionality and architecture of all living cells. B. The amphiphilicity confers lipids with wetting, emulsifying, and self-assembly characteristics. C. Lipids open a wide array of different formulations because they can be manufactured as simple one excipient solutions, suspensions, and emulsions to sophisticated multi-excipient self-emulsifying lipid system, nanostructures, and solid lipid nanoparticles. D. Lipid-based systems are commercially available to formulate pharmaceuticals for oral, topical, pulmonary, or parenteral delivery. E. Lipid formulations are modifiable to meet a wide range of product requirements as per the disease condition, route of administration, and product stability. A lipophilic drug can be formulated as lipid-based formulation when the drug itself is an oil-like substance such as ethyl isopentate, tocopherol nicotinate, etc. When conventional formulation approaches like granulation or soluble liquids in capsule do not enhance the oral bioavailability. A variety of lipid-based systems composed of simple oil solution to complex mixture of oils, co-solvents, surfactants, and co-surfactants can be obtained based on the type of excipients and formulation variables. Indeed, these systems can be converted to solid intermediates like powder, granules, and pellets by various techniques and can be filled in hard gelatin capsules or can be compressed into tablets after blending with suitable tableting excipients. Lipid-based drug delivery systems provide the vast array of possibilities to formulations as they potentially increase the bioavailability of number of poorly soluble drugs along with the formulation of physiological well-tolerated class. The development of these systems require proper understanding of the physiochemical nature of compound as well as the lipid excipients, proper characterization and evaluation of these excipients, their stability, classification, and proper consequently affect the number of these formulations. So now our first learning objective for today, lipids as excipients for drug delivery. The word lipid stems from the Greek word lipos, meaning greasy to the touch. Lipids are organic compounds of biological origin that are generally hydrophobic in nature, insoluble in water, and soluble in organic solvents. The main biological functions of lipids include signaling, storing energy, and acting as natural components of cell membranes. The term lipid is sometimes used as synonym for fats. However, fats are a subgroup of lipids called triglycerides. Lipids also encompass fatty acids and their derivatives, including mono, di- and triglycerides, phospholipids, as well as sterol-containing metabolites such as cholesterol. There are many ways in which lipids can be classified, such as A. Based on their physical state at room temperature as solid and liquid lipids. B. Based on their polarity as polar and non-polar liquids. C. Based on their charge as neutral, anionic and cationic lipids. D. Based on their origin as natural, semisynthetic and synthetic lipids. E. Based on their structure as simple, complex and derived lipids. However, in this module classification is done based on their structure, and only those category of lipids are discussed that have found application as an excipient in drug delivery. Fatty acids. Fatty acids are monocarboxylic acid derivatives of saturated or unsaturated aliphatic hydrocarbons. Usually fatty acid consists of long hydrocarbon chain C14 to C20, 14 carbon, that means carbon 14 to carbon 20. However, medium chain consists of 6 carbon to 12 carbons, and short chain consists of C4 to C6 fatty acids. Fatty acids such as stearic, oleic, linoleic and linolenic are all C18 carbon compounds and are long chain fatty acids. Amongst the short chain fatty acids are acetic acid, butyric acid are the best examples. The hydrocarbon chain of the fatty acid is often called as tail of the molecule, whereas carboxylic acid group of the fatty acid is often called as head of the molecule. The tail of the fatty acid is hydrophobic and the head of the fatty acid is hydrophilic, and thus when fatty acids are placed on a water surface, they orient themselves to form a monolayer with their head sticking into the water and their hydrophobic tails sticking up above the surface of the water. The role of fatty acids and their derivatives like fatty acid soaps, simple fatty acid ester, modified fatty acid ester and fatty alcohol either as pharmaceutical excipients has been well established. Most of the fatty acids and their derivatives are used as surfactants and emulsifier in emulsions, as penetration enhancer in topical formulations, as a solubilizer in tablet formulations, and as raw material for ointments, creams and lotions. Recently, the fatty acids have been explored as an important component of nanoemulsions and modified liposomes. Some of the fatty acids which are widely used as pharmaceutical excipients are shown on the screen for quick reference. And now we will discuss about glycerides, which includes mono, di and triglycerides, further into oil, fats and waxes. So now we will start with glycerides and its classification. Glycerides belong to the class of simple lipids, are also known as acylglycerols. These are esters of glycerol with fatty acids. Glycerol has 3 hydroxyl functional groups which can be esterified with 1, 2 or 3 fatty acids to form monoglycerides, diglycerides and triglycerides. The same is shown in the screen. Fatty acids should be either saturated fatty acids such as stearic acid, palmitic acid, etc., or unsaturated such as oleic acid, linoleic acid, etc. Now we will further discuss about mono and diglycerides. Mono and diglycerides containing free hydroxyl group are called as partial glycerides. Polar short chain partial glycerides possess excellent solvent properties for many hard to solubilize drugs, making them valuable as excipients in improving the formulation of certain pharmaceuticals. Partial glycerides are generally used as surfactants, emulsifiers and thickeners in pharmaceutical and cosmetic products. Unsaturated monoglycerides such as glyceryl monoleate, also called as GMO, and phyt and triol have been reported to form cubic and hexagonal phase and are used for the development of drug delivery carriers such as cubosomes and hexasomes. So now we will discuss in brief about triglycerides, which also forms oils and fats. Triglycerides, the principal components of oils and fats, are the most common excipients used in lipids-based drug delivery. They are fully digested and absorbed in body and do not possess safety-related issues. Triglycerides, depending upon the type of fatty acid, can be further classified as long chain triglyceride, also called as LCT, medium chain triglycerides, also called as MCT, and short chain triglycerides, also called as SCT. The capacity as a solvent for drug is mainly decided by the effective concentration of ester groups. MCT have a higher solvent capacity than LCT and are less prone to oxidation. Oils consist of high proportion of triglycerides of unsaturated fatty acids and exist as lipid at room temperature, whereas fats containing higher proportion of triglycerides of saturated fatty acids are solid or semi-solid at the same temperature. Some of the pharmaceutically used oils and fats are detailed in this module. So we will further discuss about oils. Oils are obtained from plants or vegetables called as vegetable oils, have in wide application in drug delivery. Some pharmaceutical important vegetable oils include almond oil, apricot oil, castor oil, avocado oil, borage oil, corn oil, etc. Vegetable oils contain high proportion of triglycerides of monosaturated fatty acids and polyunsaturated fatty acids along with partially glyceride materials such as lecithin and substances such as tocopherol. Vegetable oils are used as oily vehicles in the preparation of nail polish, lip balms, lipsticks, lotions, emulsions, creams, soaps, and in cosmetic preparations. These oils act as an emollient, protector, regenerator. Oils such as castor oil, sesame oil, and soybean oil have been found application as vehicle for intramuscular injections and are particularly useful for sustained release of hydrophobic drugs. Vegetable oils are used as excipients for most of the formulations, including vehicle for parenteral injections and soft gelatin capsule, as a vehicle and surfactant for emulsion, creams, lotions, and as lubricant during tablet manufacturing. Recently, there is a growing interest in the use of oils obtained from fish as vehicles for soft gelatin capsules or as oily phase of emulsion, because besides acting as a vehicle, these oils are also ideal and natural source of essential fatty acids, particularly omega-3 fatty acids such as EPA, docosahexaenic acid, which is also called as DHA, and docosahexaenic acid, which is also called as DPA. The omega-3 fatty acids helps lower cholesterol and blood triglycerides, and DHA is garnering attention as fatty acids essential to the brain and other neural tissues. Fish oils have found applications in preparation of monoemulsions, although reports are limited. So now we will also discuss about little bit brief on fats. Fats consist of higher percentage of saturated fatty acids than do oils and thus exist as solid at room temperature. The fruit and the seeds of diverse vegetable species contain fatty fractions of consistency between solid and pasty that are known as vegetable fats. The fat commonly used as excipient in pharmaceutical is oil of theobroma, also called as coca butter. It has excellent emollient properties and is used as to soften and protect chapped lips and hands. It also presents lubricating and greasing properties. It is also used as an excipient for coating tablets and in ointments. Due to its unique property to melt at body temperature, coca butter has been widely employed as a base for suppositories and pessaries. Another fat widely used as pharmaceutical excipients is called shea butter. Crude shea butter has natural antioxidant properties due to presence of tocopherols. The presence of high content of unsaponifiable fats in shea butter makes it good active ingredient against stretch marks and aging, since unsaponifiable fats keep the skin young by stimulating the tissue and helping the skin make its own collagen. Shea butter has also restructuring effects on the epidermis, anti-elastic characteristics, activity on cell regeneration, and capillary circulation. Therefore, it is highly appreciated in the cosmetic industry. After discussing oils and fats, we will also briefly discuss about waxes. Waxes are esters formed by combination of fatty acids with high molecular weight, monohydroxy alcohols. Waxes also belong to the class of simple lipids. Example of waxes used in pharmaceutical industry include Carenuba wax, beeswax, spermaceti and lanolin. Phospholipids. Phospholipids belong to the class of complex lipids, are phosphorus containing amphiphilic lipids. Phospholipids are similar to the triglycerides with a couple of exceptions. Phospholipids or glycerophospholipids consist of a glycerol backbone which is esterified in positions 1 and 2 with fatty acid hydrocarbon chain and in position 3 with phosphate group. The same is shown in the screen. There may be a variety of fatty acids, both saturated and unsaturated, in the phospholipids. Phospholipids are well-established excipients for pharmaceutical applications. As main components of cellular membrane, phospholipids have excellent biocompatibility. Additionally, amphiphilicity confers phospholipids with self-assembly, emulsifying and wetting characteristics. When placed in water, the phospholipid tail will concrete together to form a local hydrophilic environment and charged phosphate group facing out into the hydrophilic environment, forming variety of structures such as micelles, lipid bilayers and vesicles. These phospholipid bilayers and vesicles can be employed as a drug carrier. Such vesicular carriers is used for encapsulation of hydrophilic and hydrophobic drugs, protection of drug against physiological degradation, sustained release of drug and targeting of drug. Phospholipids also possess good emulsifying property and can act as surface active wetting agents to enhance the hydrophilicity of the hydrophobic drugs. These are natural compounds and effective alternatives to synthetic emulsifiers. Phospholipids are used as excipients in many types of formulations like fat emulsions, mixed micelle, liquid crystal, liposomal preparation, lipid nanoparticles and nanostructured lipid carriers for any administration route. The phospholipids can be classified by, as is also shown in the screen, A. Length of hydrophobic side chain, B. Saturation of hydrophobic side chains, C. Bonding between aliphatic moieties and glycerol backbone, D. Number of aliphatic chains, E. Variation in head group. Phosphatic acid, also known as PA, consists of two fatty acids attached in ester linkage to the first and second carbon of glycerol and a phosphate group attached to the third carbon of the glycerol. C. Similarly, phosphodicholine, also known as PC, consists of two fatty acids attached in the ester linkage to the first and second carbon of glycerol and choline attached through a phosphodiester linkage to the third carbon of the glycerol. Similarly, the term phosphodicholine is used interchangeably with phospholipid, since among the phospholipids discussed here, phosphodicholine is preferred as pharmaceutical excipient. Since then, we will discuss briefly about phosphodicholine, which is also called as lecithin. Phosphodicholine are pharmaceutically important phospholipids that incorporate choline as a head group. The term lecithin is sometimes used interchangeably with phosphofatidylcholine, although both are different since phosphofatidylcholine is one of the component of lecithin. According to USP, lecithin is described as complex mixture of acetone, insoluble phosphatides which consist of chiefly of PC, PE, PS and PI as we have discussed above. Today, lecithin has become the trade name of mixture of phospholipids typically used in pharmaceutical, food and cosmetic industry. According to the source of origin, phospholipids can be divided into natural phospholipids, semisynthetic phospholipids and synthetic phospholipids. We will now delve in brief about all the three: natural phospholipids, semisynthetic phospholipids and synthetic phospholipids. 1. In natural phospholipids, we will discuss about egg and soybean lecithin. Lecithins are extensively present in vegetable oils such as soybean, corn, sunflower and cottonseed and in bovine brain. However, egg yolk and soybean are the most important sources of lecithin. Egg lecithin and soy lecithin are different in terms of contents and species. Egg lecithin contains higher amount of PC. Egg lecithin contains long chain polyunsaturated fatty acids such as omega-6 and omega-3 series primarily arachnoic acid and doxahexonic acid, which are absent in soybean lecithins. Egg lecithins also contains SM. The saturation level of egg lecithins is higher than the soy lecithins, making them less prone to oxidation. Second, semisynthetic phospholipids. Semisynthetic phospholipids are those which are formed by changing the head or tail group or both of the natural phospholipids like hydrogenated lecithins or enzyme modified natural phospholipids. Third, synthetic phospholipids. Synthetic phospholipids involve the formation of ester or ether bonds linking apolar moieties to glycerol backbone and the attachment of polar head group. The synthetic phospholipids have the advantages of single component and stable property and are designed to optimize the drug targeting properties of lipid carriers. Carriers formed using PG allylated phospholipids have long circulation time in body, improving drug bioavailability. Utilizing liposomes constructed with synthetic lipids can create a number of novel functions. In addition to prolonged circulation time, modified head groups are useful for the attachment of cargo or specific ligand targeting. Often these modifications result in improved targeting and biodistribution of liposomes by interacting with ligands present on specific cells or tissue types. One notable example is nucleic acid modified lipids, which can result in the physical interaction of the liposomes with single-stranded nucleic acids via base pairing. Cholesterol. Cholesterol is a membrane constituent widely found in biological systems which serves as a unique purpose of modulating membrane fluidity, elasticity, and permeability. Unfortunately, cholesterol presents certain problems when used in pharmaceuticals. 1. High purity sources suitable for clinical applications are not available. 2. Commercially available cholesterol is derived from egg or wool grease and is not suitable for pharmaceuticals due to potential viral contamination. 3. Cholesterol is readily oxidized, creating a stability problem for lipid-based drug products. Further, the oxidation byproducts are toxic in biological system. Despite these disadvantages, cholesterol has been widely employed with other lipids during manufacturing of vesicular liposomal formulation, lipid nanoparticles, and lipid nanostructures. Third learning objective is properties of lipids as pharmaceutical excipients. The species diversity of lipids and selection of lipid to achieve the therapeutic purpose become a crucial problem in the design of lipid-based drug delivery system. So we summarize the property of lipids which can give a guidelines in the design of lipid-based drug delivery systems. So mainly we will discuss about 5 properties. A. Absorption and digestion of lipids. Formulations developed using lipids as excipients can reduce the inherent limitation of slow and incomplete dissolution of poorly water-soluble drugs. If the drug possesses high affination to the lipid vehicle, it can assume that the drug moves apparently together with the vehicle in the GI tract, revealing that the digestibility of lipid would be important for drug absorption. Thus, careful selection of lipid vehicle can control the absorption rate of the drug. Lipid-based system increase absorption from the gastrointestinal tract by A. Accelerating the dissolution process, B. Facilitating the formation of solubilized phases by reduction of particle size to the molecular level, C. Yielding the solid state solution within the carrier, D. Facilitating transcellular absorption due to increased membrane fluidity, E. Allowing paracellular transport by opening tight junctions, F. Increased intracellular concentration and residence time by surfactants due to inhibition of PGP, G. Lipid stimulation of lipoprotein production, H. Enhancing drug transport to the systemic circulation via intestinal lymphatic system. Although most orally administered compounds gain access to the systemic circulation via the portal vein, lipid-based systems may be transported directly to the systemic circulation via intestinal lymphatics. Some of the advantages of lymphatic transport of drug are avoidance of first pass metabolism, improved bioavailability, and targeting of specific diseases which are known to spread via lymphatics, such as certain lymphomas and HIV. Second property, polymorphism and structural organization in water. Lipids can form many kinds of assemblies in water such as hexagonal phase, cubic phase, micelles, and liposomes attributed to their molecular shapes. Understanding the lipid polymorphism is essential for developing the stable carrier and triggered release lipid-based delivery system. Non-bilayer lipids such as glyceryl monostearate and unsaturated phosphodiethylamine can be stabilized in a bilayer structure by the presence of bilayer preferring lipids such as phosphatidylcholine, phosphatidylcerine. Usually 20 to 50 ml molecular percentage of bilayer preferring lipids are required to maintain a bilayer organization when mixed with hexagonal phase forming lipids such as unsaturated phosphatidylethanolamine. The structural preference of these lipid systems can be modulated by a wide variety of factors such as hydrocarbons unsaturation, head group size, pH, ionic strength, and temperature. The same is shown and explained on the screen. When exposed to aqueous environment, various types of amphiphilic lipids spontaneously form thermodynamically stable self-assembled structures and eventually develop into cubic or hexagonal liquid crystals depending on temperature and water content. The liquid crystal forming lipids are non-toxic and biodegradable and can be used for various routes of administration. Such cubic and hexagonal liquid crystals have received considerable attention because of their potential as drug delivery systems. Cubic and hexagonal liquid crystals provide a slow drug release matrix and protect peptides, proteins, and nucleic acids from chemical and physical degradation. Now coming on to the third property, C, phase transition temperature, also known as Tm, as shown on the screen. The phase transition temperature of a lipid is defined as the temperature at which lipids transit from ordered gel phase, where the hydrocarbon chain are fully extended and closely packed, to disordered liquid crystalline state, where the hydrocarbon chains are randomly oriented and fluid. There are several factors which directly affect the phase transition temperature as also shown on the screen. Number 1, the hydrocarbon chain length. Number 2, the nature of the polar head group. Number 3, the degree of saturation of the hydrocarbon chain. Fourth, purity. The proper understanding of transition temperature of the lipids could be useful for the development of stable and robust lipid formulation. Choosing a high transition lipid where the lipid vesicle would always be in the gel phase would provide a non-leaky packaging systems. Alternatively, a lipid with a transition temperature between the starting temperature and the ending temperature of the system would provide a means of releasing packed material as the lipid passes through its phase transition temperature and the vesicles become leaky. Also, one should consider how the transition temperature of the lipid could impact the processing steps. During manufacturing of liposomes, hydration of lipid layer should be done necessarily at or above transition temperature in order to avoid processing problems. Coming on to the fourth property, charge. Naturally occurring lipids are either neutral such as PC and PE or anionic such as PS, PI, PG and PA. However, cationic lipids are prepared synthetically. The head group of the synthetic cationic lipids typically comprises various amine derivatives. Various amine head groups and the respective examples are summarized as follows, as shown in the screen. The fifth property, E, stability. The shelf life or long-term stability of a drug product containing lipid as excipient can be dramatically affected by the lipid species used in the formulation. Generally, the more unsaturated a compound, the easier the product is oxidized and thus the shorter shelf life of the product. Lipids from natural resources such as soya bean or egg typically contain significant levels of polyunsaturated fatty acids and therefore are inherently less stable than their synthetic counterparts. While saturated lipids offer the greatest stability in terms of oxidation, they also have much higher transition temperatures and thus present other difficulties in formulation. Now we are moving towards the end of the module, and the last learning objective of the module is lipid formulation classification system. This was introduced as a working model in 2000. In recent year, also called as LFCS, have been discussed more widely within the pharmaceutical industry and is adopted as a framework for comparing the performance of lipid-based formulations. The main purpose of LFCS is to facilitate the identification of the most suitable formulations for specific drug with reference to its physiochemical properties and to enable in vivo studies to be interpreted more readily. LFCS classify the lipid formulations into 4 classes on the basis of composition, dispersion and digestion characteristics. First, type 1 formulation consists of lipophilic ingredients like oils that are not dispersible in aqueous media but have the desirable properties of being rapidly digested and absorbed completely from the intestine. Second, type 2 formulation consists of oils and water insoluble surfactants that form fine oil-water emulsions when introduced into aqueous media. They are likely to be digested. Third, type 3 formulations are well dispersible, formed with the help of co-solvents such as propylene glycol. Digestion is not needed for these formulations. These are subdivided into 3A and 3B systems. The 3A system consists of water-insoluble surfactants, whereas 3B consists of water-soluble surfactants. Fourth, type 4 formulations contains no oil. They are mainly composed of water-soluble surfactants and co-solvents capable of forming micelles on contact with physiological media but having limited or no digestibility. So lastly, now we will conclude the module. A wide range of lipid excipients are available from excipient suppliers. Since these lipids affect the absorption process, it is necessary to know the characteristics of various excipients. The factors that determine the choice of excipients for lipid-based formulations include miscibility, solvent capacity, self-dispersibility and ability to promote self-dispersion of the formulation, digestibility and fate of the digested products, regulatory issues, irritancy, toxicity, purity, chemical stability, capsule compatibility, melting point and cost. In most of the cases, a single lipid species does not yield the particular physical properties needed for a delivery system or does not adequately mimic the natural system for which it is needed. A lipid mixture composed of 2 or more individual lipids usually serves the purpose. As for example, usually mixture of lipids are needed for the formation of liposomes, cubosomes, solid lipid nanoparticles and nanostructured lipid carriers. On the way of conclusion, the prospect of these delivery systems looks promising. Thank you.